Hi, everybody. Good afternoon. Thank you, Lindsay, and thank you to ARM for the kind invitation to come and talk to you about Homology Medicines. We're very excited about what we're doing. It's very exciting to see everybody in this field exploding like it is, and it's great to catch up with friends and colleagues. We are now a public company, so there are forward-looking statements, and if you want more information, you can check out our SEC filings on our website. So this is really, I think, why we're all here, and we're at a point in time now where the science and medicine has evolved, where we can actually consider curing these patients with rare monogenic genetic disorders and potentially larger diseases. And this is what drives Homology, drives many of your companies. This is the touchstone for us and the passion that we bring to bringing this platform to patients. Homology was formed about 3 years ago around a discovery from the City of Hope from Dr. Shastri Chatterjee's lab. And 5AM Ventures and Arch Ventures were able to put together a company with a team of former, initially Shire colleagues with a lot of experience in developing rare disease drugs. So we've known how to do this from taking something from a basic research perspective all the way through the clinic, all the way into commercialization. And if you look in the lower left-hand side, this discovery was essentially the discovery of 15 novel AAVs that were isolated from human hematopoietic stem cells. So we term them AAVHSCs. And these all map to clade F along with AAV9. And AAV9 actually was also pulled out of these stem cells as well. So they're very similar to AAV9, but have distinct composition of matter around them. And they have the ability to highly and efficiently mediate gene transfer, but also have the ability to do homologous recombination-based gene insertion. And this means that we're able to do this, we and others are able to do this without making double-stranded breaks in the DNA in order to insert genes. We will be in the clinic next year in the upper right-hand part of the slide with our lead program in adult PKU patients. With a gene transfer approach, and we will have data, at least the initial cohort of data, in 2019 as well. So we've articulated that timeline for the last 3 years, and we're still right on that timeline. Manufacturing is really a big part of this, and we've been able to manage that as well, and broad intellectual property here around these AAVHSCs and methods of use and composition of matter. This is a team. These are mugshots. I'll point out Albert Seymour, who's Chief Scientific Officer. He's really the brains of the operation. He's a human geneticist by training, but with a lot of drug development experience. Sam Rosti, Brad Smith, our CFO, with a lot of experience in emerging growth companies. Tim Kelly, who heads up our technical operations, former Biogen, Sarepta, Shire head of tech ops, a lot of experience, and Teresa McNeely, head of corporate communications and patient advocacy. Great team. So this is essentially the platform. It's a bimodal platform. On the left-hand side is your basic gene transfer construct within these AAVHSCs. So what you see are the ITRs with a promoter, constitutive promoter, or in some cases a organ-specific promoter with a gene of interest, and that forms an episome. And we see excellent biodistribution properties around these AAVHSCs, and we have the ability to select one of these 15 that goes to the tissues where we think we need to get to. But what makes this platform interesting is on the right-hand side, if you take that same AAVHSC and you have now a single-stranded DNA, cDNA construct with homology arms on either side of the correct gene, those homology arms guide specifically and integrate into the chromosome behind the natural promoter, if that's the way we want to target it. So you can permanently make that correction, and that flipping in of the correct gene, flipping out of the mutated gene, and make that permanent correction to the genome, which is now passed down to daughter cells. So this is really, we can let the biology of the disease dictate whether we use the left-hand side, so if you're in slowly dividing or non-dividing cells, adult liver, the CNS, you can go with the gene transfer approach and hope for a long-lasting solution. Or on the right-hand side, this is a methodology where you can go after diseases where the tissue is rapidly dividing, for example, the bone marrow or the pediatric liver. This is the way we think of ourselves in terms of how we position and what we can go after, what we do well. We can insert genes very nicely, precisely, with little to no off-target into the genome. So we can really go after autosomal recessive disorders, which comprise the majority of rare monogenic disorders. So you can see in that lower pie chart that about 70% of all the monogenic diseases are autosomal recessive, and that's because both of these alleles have the mutation, and you basically got to kick that gene out and put the correct gene in. If you have a technology that makes a double-stranded break, going after autosomal dominant diseases makes a lot of sense because there you just need to cut one of those alleles, the gain-of-function allele, in order to affect a cure. So we feel like we're very well positioned to go after autosomal recessive. This is our pipeline, and you can see our strategy was to start with gene transfer first, which is a well-known now regulatory pathway the FDA is much more familiar with thanks to all the pioneers in the field that have gone before us, and to go after the adult PKU population, and this is one of the more common rare genetic disorders. And with the idea of starting this trial next year and having data next year. Behind that, we're going to go after a CNS disorder, which is a lysosomal disorder, metachromatic leukodystrophy, with an in vivo approach, taking advantage of that the brain cells don't divide, so you can do gene transfer there. And as long as you have a highly efficient transduction rate in the tissue you need to get to, in this case the white matter, then you have a very good chance to impact this pernicious disease where these kids die by the age of 10. In gene editing, we're going to go after the pediatric population of PKU, which are identified by newborn screening every year. And there's about 300 to 400 patients that are identified just in the US alone. So because the livers are quadrupling over the lifetime of a child, you want to be able to make that permanent correction. And from a regulatory point of view, this strategy is kind of a 1-2 punch where we'll have a lot of safety data around the vector itself in the adult population and use that same vector to go after the pediatric population in children. So these kids lose IQ points and they lose executive function over time despite being on a very strict diet. So here we can really intervene at a very young age. And behind that, we have a couple of hemoglobinopathy programs because these vectors home very nicely to CD34. And we partnered with Novartis last year on sickle cell, which is an in vivo approach for sickle cell to put the beta globin gene in and make that permanent correction. And they wanted to go after a couple of ophthalmic targets, which is great because they have the expertise there. It's not in our wheelhouse, but it leverages our platform to bring forward for patients with these genetic eye diseases. So one of the things about PKU is there's 2 drugs on the market now, Kuvan, Palynziq from BioMarin, that are really suboptimal, and they're bringing forward a non-natural enzyme, phenylalanine lyase. You can see on the right-hand side of this slide, you're basically taking phenylalanine and you're degrading it in the blood, and this is an extracellular enzyme that's pegylated in the case of Palynziq, and you're turning it into tranexamic acid and ammonia. So these are non-natural metabolites. What we're doing here in this case is we're putting back PAH, or the phenylalanine hydroxylase enzyme. And that is the natural enzyme that these patients are missing, and that converts phenylalanine to tyrosine, and tyrosine is an important precursor for all the neurotransmitters, dopamine, serotonin, et cetera. So that's the concept here, is to really put back the naturally occurring enzyme with the hope of lowering phenylalanine to the levels that these patients can eventually go off diet, but importantly also generating the neurotransmitters so you can have the ability to go after neurocognitive endpoints that the BioMarin products haven't been able to hit. So there's about 15,000 patients in the US, 50,000 globally, and again, newborn screening is identifying these patients every year. This has been since the mid-1960s, and the incidence of PKU has been the same for all 55 years now. And so here is our preclinical data. There's an excellent mouse model, the ENU2 mouse. This is used to translate Kuvan and Palynziq, where Palynziq showed about a 50% reduction in phenylalanine, and that's about what they showed in the phase 3 trial. But here, if you look at the top blue, the left-hand graph, top blue bar, what you see are normal Phe levels in this mouse, and it's pretty much at 1,200 micromole, which is where you see the classic or more severe form of PKU patients. With a single injection of AAVHSC 15 with the PAH gene, that drops down to baseline after 1 week. And we have these data out now past 42 weeks, which is showing a durable effect here long term. And importantly, on the right-hand side, what you're also doing here is normalizing the Phe to tyrosine ratio. That bottom line shows that it's about a 1-to-1 ratio, and that's where you want to be. We're actually making tyrosine, and that's the big differentiator here with this approach versus any approach that uses the PAL, or phenylalanine lyase. Also for editing, this is early data, initial construct coming from the City of Hope manufacturing process. This is about 4% editing in the alleles of the ENU2 mouse. And you can see here we dropped the level of phenylalanine by 50%. That's pretty much where Palynziq was in its phase 3 trial. We have now converted this process to a GMP process, scaled this up, and with that purification level at GMP, we are seeing, and with optimizing the homology arms, we are seeing now that editing walk up to that magic 10% level. That's the level you need to get to. So that's data that we'll be showing in the very near future, but for now we have to show you this, but I can tell you it's going to get better. Great thing about using AAV-mediated HR is that you're taking advantage of a natural DNA repair mechanism, which is known to be highly precise. So Albert's team has really gone to great lengths to develop an unbiased method to detect any off-target. And we don't see any off-targets within the era of the NGS assay itself. There's no integration of the ITRs, no indel formation, and just where we're wanting to go, we find it, and it's nowhere else in the genome. And that's exactly how HR works, and that's because you have these long stretches of nucleotides that form the homology arms on either side of that gene, which allows it to be extremely precise. As I mentioned earlier, these vectors have very good biodistribution. We did non-human primates very early on to see where did these vectors go. We looked at a number of these AAVHSCs, and the nice thing is, on the right-hand side of the slide, you can see we have vectors that can go to multiple tissues very well to affect and impact the disease. For example, in Friedreich's ataxia, you want to hit the dorsal root ganglia, the dentate nucleus, and the cardiac tissue. We have a couple of our vectors that hit all three of those tissues, so those would automatically jump forward as candidates for us to develop a therapy for a disease like that. We have moved along really quickly on the manufacturing front. Very early days, we started right out of the gate. We've got a lot of battle scars from our Shire days in manufacturing. And so this was a huge focus for us. We've invested now in a phase 1/2 GMP facility. This will be done by the end of this year. It's 25,000 square feet, which we'll be able to be moving forward with. And we will grow this internally as the company grows as well. And I think manufacturing and controlling that is a huge advantage from a strategic point of view. We already are at scale for our phase 1/2 in gene therapy for adults, at scale and at GMP in a 2x200 liter system, and we're using HEK 293. So we really have this, I think, in a place where we feel good about being able to move forward, where we can make multiple lots to basically take this whole trial all the way through. So last slide, where are we now? So just from a short 2.5 to 3 years ago, we're going to begin this trial next year. We're already selecting our sites, talking to KOLs. There's definitely, and talking to patient advocacy groups, there's definitely a need and a want for something that's a one-time treatment that represents a cure for these patients versus what they're taking now in terms of diet and other drugs. We will nominate by the end of 2018 a gene editing candidate that will move into IND-enabling studies in 2019. We'll complete build-out of our phase 1/2 facility by the end of this year, and we're continuing to advance our pipeline along with MLD, and we'll have more news on that as we go along, as well as our programs with Novartis in ophthalmology and sickle cell and hemoglobinopathy. And that's the story. Thank you very much for your attention.